Thank you for having me. And today, my talk was about the fluid physics-mediated evolutionary convergence in behavior among marine plankton. So, I mean, convergent evolution in the fluid biofluid mechanics has not been a new thing, and it has been. I mean, the earliest, I think the earliest example that people talk about is the hydrodynamics and the convergent evolution in fast-swimming fish-like animals. So by definition, convergent evolution manifests as a pattern in which similar phenotypes evolve independently in multiple lineages. So in his book by Lighthill, Sir James Lighthill, he said that the goals are not just to understanding the functioning, but also to understand the evolution of fish swimming. So morphologically, you see that there is a I mean there are many species of fast swimming fish. They all evolve into a lunate tail, and also the body necking anterior to it, something like this, and including actually an extinct animal. I mean extinct animal, and we can guess that the behavior, because of they have a similar morphology, so the behavior should be similar to this. So by behavior, we see that all these fish, these fast-swimming fish, they actually swim in a carangiform, all the tuniform swimming modes. Basically, it's a tail beating like this, and the tail beating generates a vortex, inverse von Karman vortex strait that generate the thrust. And for this one, actually, the convergent evolution was played by the high Reynolds number flow. So this is I just want to give here as an example, early example, and to introduce the convergent evolution in the fluid mechanics context. But today, our talk will be mainly focused on the smaller, much smaller sized animal, the marine organisms, which are, say, if you see from here, that's the marine food webs. Basically, they are consist of many smaller, much smaller organisms. Say you see that here's the fish, then this is the food chain by the large zooplankton such as copepods and also the algae. And from here you see there consider a lot of smaller sized organisms, especially those here is called the microbial loop. I don't want to talk about the details about these food webs, but just want to emphasize that in the marine organisms, that the smaller than 1 millimeter organism, the size smaller than 1 millimeter, they contributes to 99% of the biological activities in the ocean. So I mean, this can be demonstrated by this Shelton's paper, and also most updated one is by Houghton, which show that the global ocean size spectra, which are mostly you have a large amount of smaller organisms that are abundant and contribute to the biological activities. So I mean, here I just want to say that in the ocean, we are dealing with all these very small organisms. So how fluid mechanics play in their life, and is a topic of my talk. So also I want to introduce something called the key taxa. So this is by the paper by Verity and Smitacek in 1996, and they say that the key taxa have strong environmental adaptations, whether through behavior, morphology, physiology, or life history. And they direct trophic relationship and spearhead biogeochemical fluxes. And there is a need that we need to study this dominant key taxas behavior, the life history, behavior, morphology that account of their size. And for me, over the past, I think, probably 15 years, I have been able to study many of those I consider the key taxa organisms, marine organisms. So So what I do is that I actually observe their behavior and study their fluid mechanics, how the fluid mechanics affect their life. And so I can see that from here, you see these copper pots, and also these ciliates, and also this is heterotrophic and mixotrophic dinoflagellates, and also larvae, copper pot larvae, and also larger things like cryomycetes, and also smaller things like cryptophytes. You see the size span like this. So I actually observe, I mean, collaborated with many collaborators, and I have actually observed the behavior, the individual level behavior of all those animals, all those organisms, and I can see that there are some patterns which are convergent evolution, which actually can show to demonstrate that. So I mean, these are many terabytes of movies, which I think is very interesting, and I just want to report some of those results related to those key taxa. So my research tool is actually what I developed is called a high-speed microscale imaging system. So this system can be conveniently to conduct quantitative microscopy in the high speed and high resolution fashion. And basically it consists of a high speed camera, which can take movies at 1,000 times 1,000 pixels up to 2,000 frames per second. And also it has a long working distance long working distance setup, and can observe a relatively larger, say a smaller water vessel, which we can hold the organisms inside. And this is basically much better than you just observe under a microscope, because I think the traditional microscopy has several limitations. For example, they confine to a horizontal field of view, and here we use a vertical field of view, and also wall effects due to small sample volumes. Here we use a relatively larger tank to observe it, and also the traditional one has a strong convection caused by strong lighting, and we use a very weak 1 watt LED lighting. So for this one, we emphasize that to observe these smaller organisms, you must using a very high speed in order to be able to see what they really do. So this is my research tool. So next, I want to talk about something about the regimes of the fluid physics for marine organisms. So here you see the first one is obviously our Reynolds number. So it's a UL over nu with, so I choose U equal, the U is the velocity of the swimming velocity, which is about I choose 100 body lengths per second. You see that in most of this size range, the Reynolds number is a kind of small, means the viscous effects are dominant, or at least cannot be neglected. And also the second one is the Peckley number, which are combined to the fluid transport versus the fluid the diffusion process. And the same thing I choose, you can see that there are some ranges that the fluid number, the Peckley number is small, and also there could be a relative larger one. So what we really interested is the time scale of it. So the time scale has two time scales. One is the viscous time scale, which defined by the L, the length divided by the viscosity. And you see that this viscous time scale is relatively smaller than 1 second or across all these ranges. And the next one is the diffusion time scale, which is I mean, just replace the nu with the diffusivity. You see that also at some range, this diffusion is also very small at a smaller size, and there could be a relatively larger. I mean, at like 100 meters, depends on what you choose with the parameters. But the thing is because we have these two physical physical parameters, so the organisms can do something, which they can choose to do something fast. So the first thing, if they can do something faster than the diffusion time scale, which is in this range, I just say the first range convergence could be shown here. That is, if the animals, the organisms, they can behave faster than the diffusion time scale. So this is the first category that I'm going to talk about. So my example is that the first example is the photosynthetic bloom-forming ciliate. Is called Mesodylium rubrum. This is actually a ubiquitous species complex. So these things is this species actually has like a spherical body shape and have a cilia which at the equatorial side, and this species actually has the highest productivity ever measured for phytoplankton blooms. That is achieved the largest carbon fixation rate, and also they form red tides that are biogeochemical hotspots. And also, I mean, as I said, it's a species complex because of recent genetic studies of those Mesodylium populations. They actually demonstrated that they have cryptic diversities. That means the DNAs could be different, although they behave or the shape similarly. So it's a species complex. So here you can see that they generate worldwide red tides, and here example at the red tides generated at the Columbia River estuary. These red tides. So this jumping this ciliate is actually a jumping ciliate. It's called so I can show how by doing high speed micro scale imaging, you can see how they actually jump. So here is an example of how it jumps. So basically, how it jumps is that it beat this equatorial cilia belt backward. Then you see this is the oral tentacle. So they bring this is actually the oral parts. So they jump backward. So then they jump forward like this. And the jumping distance can be like, say, for this specific one is about 132 micron. And the jumping speed is about, say, this is jumping speed time and jumping speed. The largest one can be 7.9 millimeter per second, 8 millimeter per second. So this is compared to like a 30 micron size. So this is that's a huge jumping speed and a jumping distance. So I have been working with Matt Johnson at WHOI. So we study the jumping Mesodylium, the species from different part of the ocean in the world. So Matt Johnson actually has species which are collected from Antarctica. So it's at 4 degrees, and also he has a temperate species which are collected around the WHOI area. So we basically do experiments. We measure their maximum jumping speed for the Antarctica one at 4 degrees. And we also force the temperate species at 4 degrees, and also the temperate species at 20 degrees, but with blue light, and also temperate at at 21 degree at white LED light. You see that those maximum swimming speed, the jumping speed varies for different species, at different conditions. And also the total jumping time also changes because of, for all these things. But what they didn't change is the body's length specific jumping distance is all about 6 body lengths. So it's for all those conditions and the different species. And we also find out that this the total jumping time scaled by the diffusion time scale, you see that they all relatively smaller than the diffusion time scale. That means they can overcome the diffusion time, the diffusion process. So they are faster than the diffusion process. So also you see that the jumping distance is all about, has a peak at about 5 to 6 body lengths. So those things actually suggest that although those different species at different conditions, they, you know, they jump at different speed, they jump different time, but their jumping distance are uniformly at 6 body lengths. So that means that they just jump, the distance is the same. So why is that? So there is a, if we goes to the diffusion limitation of nutrient uptake, these concepts, say if you have an algae, then you have this, say they build up around themselves because of diffusion process, say they generate a diffusion boundary layer. So like this. So this is the scale of the diffusion one. The highest side here has some deficit of the chemicals that has a diffusion boundary layer. So say if this is a nutrient, so in order to refresh this nutrient around this cell, and the cell has to jump out of their own boundary layer. So how much they can jump in order to renew the fluid around them is actually, you can see that this is about 6 body lengths. So each time if they want to jump to renew the water around their body, the way they do is they have to jump by 6 body lengths. So this is actually what we see from all this species complex of the Mesodinium jumping. And the other things I think is also the fluid mechanics played is why this morphology of the Mesodinium rubrum, they actually possess a equatorially located proportion ciliary belt. So they are actually at these equatorial ones. So here is the CFD simulated flow of a jumping ciliate, but consider different morphological ciliary morphological morphology. So one for this is a Mesodinium which has a equatorially distributed cilia around here. Then this is the puller. So puller means at the head part there is a cilia, so they pull forward, and the pusher is at the say the tail part, they push the cell forward. You see that if they swim at exactly the same speed. So here is the jumping ciliate impose the flow field. So the Mesodinium rubrum type, you see that the flow disturbance are actually small compared to all the other two, pusher and the puller. And the decay rate is higher, so 1 over r cubed, and the other one is 1 over r squared. So you see that by having an equatorially located propulsive ciliary belt, the ciliate Mesodinium can jump relatively fast, relatively more efficient because of they have a relatively larger fluid propulsion efficiency. Say this one versus these two, right? They're larger fluid propulsion efficient means that they're more efficient. Also, the flow, the disturbance they generate are decay fast. That means they are safer because of other fluid detection predator will have less likely to detect the presence of them. So this is also, I see them as a convergence because this is a species complex. They just converge to those kind of jumping behavior, say, and also the jumping morphology. So for achieving better the capability to do the diffusion against the diffusion process, also being efficient as jumper. So this is the convergent evolution, which are expressed forced by the fluid mechanics and expressed by those Mesodinium rubrum species complex. The other one I want to talk about the divergent evolution because of you see for those Mesodinium, they have the family of Mesodinium has quite a few different species. The different species say this is a Mesodinium major, which are very round and big. They are mostly phototrophy, just like a Mesodinium rubrum. They do a big, good photosynthesis. And then Masodium rubrum, then Masodium chameleon, and the pulex, and the pupula. And from here, they become heterotrophic. And the shape changes a little bit. So what we see from doing all those observations, we see that when they are being fully phototrophy, they actually jump very fast. So jumping speed are relatively fast because of they need to overcome the diffusion limitation. And also the jumping distance is about 6 body lengths per jump at this range. But when the species become more heterotrophic, they actually jump slower at a relative irregular jumping distance. And also, say the Mesothelium is a almost phototrophic. They still need to capture to feed about 1 cell per day. So the feeding process are very interesting because of they are actually phototrophy. They're feeding, so the capture behavior are very different from all these heterotrophic jumping cell capture behavior. So I can show here the difference. So here is the how the Mesothelium capture. So these are real time. So you basically cannot see much because of they just poke the cell this. But here we can see something like when they are, this is 20 times slower. So it's actually an ambush feeder. So they wait for a cell to swimming around, then they poke it. So within 1 second, they actually poke this poor cell about 17 times, and in order to immobile it. I mean, this process of capturing is sounds like very slow and not efficient to me. But why for the heterotrophic ones, like this pulex, Masodium pulex? So this is also an ambush feeder. So they wait the cell to be close and they detect it. Just one poke, then they immediately be able to ingest it. So this is the capture process are much faster and efficient. And here is the real time. So the real time, you basically, I mean, instantly you capture the cell immediately. I mean, within like maybe 100 milliseconds, the capture process would be finished. But here it takes much longer. So I see that there is some kind of divergent evolution because of the trade-off between jumping capability and the pre-capturability. There is a trade-off there because of the different their different capabilities in order to achieving different life process. So here. So the next one, what I want to talk about is the if the time scale of the motion of the organisms, which are larger than the diffusion times, the viscous time scale. That is here. So this is another group of animal organisms. They show different things. So here is the example of the example 2. So what I want to show you that is that all those organisms, they have impulsive jumping, which are fast enough to overcome the viscous process. So I give 5 examples of those organisms. So first one is the paralarva of squid. So you see that they jump, they generate 2 symmetric vortex ring. One is at the body side, the other one is at the tail. I mean, just push away. And the second one is the Canonus phimachicus. They also kick the same thing, viscous vortex ring back and head back. Head forward. And this one is a jellyfish, a small jellyfish. They also do the same thing, push a viscous vortex ring, and the body itself become a vortex ring too. Then the next one is the Acacia tansa, which the jump also generate 2 rings. One is the body is bound, the other one is the tail pushed away. And here is another one is a Pseudotontonia ciliate. it generates a ring forward and backward. So all those 5 different organisms, they are very different in terms of shape, in terms of, the different morphology for jumping. They actually all generate viscous jumping induced viscous vortex ring. So here is the viscous ring for the squid, and this is the copepod. The Canada's Phimachicus have a head ring, body bound ring, and also a viscous ring which pushed away. Then this is small jellyfish too. So all these, you see that there's some symmetric between the body vortex and the wake vortex. And this actually, you see that I think they imply a hydrodynamic camouflage for the jumping organism, because for a predator who detect this flow, they may have difficulty to pick up which one, here or here is the organisms which are really aside resides. And also, this viscous vortex ring decays very fast. So it's a viscous vortex ring detect. This is the decay of the circulation, which is a circulation feed to different fluid mechanics model. So what I say that, because of this viscous ring, they can give the organisms some kind of camouflage because of the symmetry between the body vortex and the wake vortex. Also, the flow decays very fast. Within like a few minutes, 100 milliseconds, it decays. So those kind of jumping is actually very safe, or also energetically efficient. So I think this is why these different organisms, they all converge to do the same thing. This is another example of the evolutionary convergence. So, here I give the fluid mechanics explanations. So you see that, this is two fluid mechanics solutions. One is called a Stokeslet, the other is a Stresslet. So I just want to show that this two flow actually decays very fast. One is 1 over r cube, 1 over r force. So the Stresslet probably decays the fastest flow field that organisms can actually generate. And this is exactly what these organisms, they do. And here I want to give a very simple non-dimensional argument about this process. So here, this is the kinematic, the kinetic energy of the vortex, and here is the impulse, and here is the circulation. And you see that this parameter, non-dimensional kinetic energy of the vortex, therefore the Stokeslet and also the Stresslet, these are very tiny, small compared to the Hill's vortex, which is a potential vortex ring. So this viscous vortex ring, actually, they are energetically less costly to generate for the same amount of hydrodynamic impulse than the Hill's vortex ring, which is like a fish generates. So you can see that by using this kind of viscous vortex ring, you can be highly energetically efficient because of you generate a high fluid propulsion efficiency, and also because of it is a viscous vortex ring, it decays faster. It make the organisms safer because of they just decay faster. So the predator who can detect those flow, they have less time to actually detect them or trace them. So this is I feel that this is actually evolutionary convergent that are driven by this viscous vortex ring fluid mechanics. So the next one I want to show that all those I have a jumping number, which is the propulsion time that is performed by the organisms, defined by divided by the viscous time scale. You see that for all those organisms who do those viscous vortex ring things, the jumping number is actually smaller than 1. They are faster than the viscous process for all those different Reynolds number range. And here is the Mesodinium. The Mesodinium actually, they operate in a different regime. That is, their jumping time are they're longer than the viscous time scale, but they are larger than they are shorter than the diffusion time scale. So you clearly see there are two clusterings. One is the Mesodinium type, the other one is those things. So these are two convergent points for all the marine or for all these smaller marine organisms. So the third example, what I want to see that is for those different size range of organisms, like marine organisms. So first one is the mesozooplankton, which is a 200 to 220,000 micrometer in size, like a copepods, right? So the copepods are very important. So I just emphasize here, and they have three different feeding process. I mean, this is all relatively familiar to us. One is the feeding current feeding. So here's the Eurythemera is feeding on these tiny Alexandria cells. And here is the Classophocatus, Classocanatus foccatus is a very fast swimming cruising field feeding process. And here I want to show how they feed. So I mean, this is a very slow down a movie, but you see that when they approaching here the cell, they actually has to stop then to capture it. And I mean, this is a milliseconds. You see that that's actually pretty fast, the swimming speed. And the other one is the Oisona plumifera, is ambush feeding. So here is a cell. Then they goes here, then they just ambush feeding out. I mean, I think we relatively, I mean, in this community, people relatively familiar with these copepods. They do these three different types of feeding mode. But what I want to emphasize that it actually not familiar for the microzooplankton. And the microzooplankton, so basically here I'm talking about the ciliates. And the marine plankton ciliates, they actually have three major groups with different morphology, swimming behavior, and the feeding mechanisms. And they were considered, traditionally considered field feeding. I mean, by Professor Tom Fenchel, who is the basically the very beginning, he studied a lot of these things. But he said that it's a field feeding. Although people later on, they think that this field feeding probably not that right, but the details, how those ciliates actually they feed was not very clear. Because of the field feeding, they was actually originally proposed by studying some benthic ciliates. So here, what we have done here is to study the dominant group of the planktonic ciliates, which are basically have three types, dominant types. So it's a Coreotrix and also Oligotrix and the Cyclotrix. So here, I mean, the Coreotrix, what here is that the ciliate actually has a circle around their oral parts, a complete circle. So they have a tintinnates and all loricates. So here has a loricated. So we found that these loricates and the tintinnates, they actually are feeding current feeding. So I can show the movie here. So you see that clearly this cell was actually pushed into by the feeding current that generates, then take it. And also here, these are loricates. That you see here, there's a cell here. Maybe I can make it faster. So you see that actually they generate the general feeding current to capture it. And for the oligotrix, so what oligotrix is that they have a incomplete ciliary circle, which is you can consider as a corner, and also a ciliate belt, which are like a lapo. So like a corner and a lapo arrangement of the ciliates. And there's a question actually in this community and the ciliate morphology community that why some ciliates has these circles and some doesn't have. And here we actually see that. So here is the feeding. So for those oligotrichs, they're actually swimming fast forward, that they push a particle there, then they use a reverse to capture it. So you see that for these oligotrichs, when they try to capture ciliate particles, they need to stop in order to accurately capture it. How they achieve this stopping motion is that because of they have this leper ciliate belt. They swimming, they are in action when they want to stop. It's basically these things actually are brake. Is a brake function. Is a brake function. So here we actually our observation first explained why some ciliates has a leper the colon leper arrangement, as some other one has a different one because of they have a different feeding mode. So this is a cruise feeding because these things are swimming very fast, just like the copepods would do, the Clausocanalus does. Then the next one is the cyclotrix, which is the ambush feeder that I have already showed before. So what I here want to talk about for this different size range, I mean, just one order of smaller one, they actually display the same dominant feeding modes as the copepods. And why is that? Because over here, they facing the same fluid mechanics constraint. Also, the very dilute feeding, the food environment, very dilute ocean feeding food environment. Also, they need to play with the predator prey and the particle encounter. So by doing that, you actually see that they converge into these three different feeding modes. It's all because of fluid mechanics related things. So the final example I want to give is the swimming behavior of copepods, which are swimming and sink behavior. So for many different species of copepods in the coastal area, actually they display this swimming and sink behavior. So what we see here is that the copepods, when they swimming, they just say swimming upward, then stop swimming, then sink back to the same position they started the swimming. So by repeating doing those things, and and what they do, I mean, why? I mean, this is many people has observed such a behavior, and the question is why they do that, and I think there's only one attempt that try to understand this kind of behavior in terms of in terms of mechanical energetic efficiency. And I mean, they actually claim that by doing that, they actually save energy. So if you just do swimming up, then sink back, you actually save energy by just hovering there. So, but my own computational fluid dynamics study, which show that this is not the case. So actually, if you do the swimming and the sink behavior, you actually are energetically more costly by doing that. And here this is actually so I actually disprove the other people's hypothesis. So I need to have a alternative hypothesis to support my thing. So still, why they do that? And my observation show that this kind of behavior actually is good for the organisms, for the copepods to feed on diatom chains. So let me show how it works. So here is a late copeptides of centropages in swimming sink behavior. So I can show how they do. So first sink, then immediately approach here, they probably detect this ciliate diatom chain, then they just sink back. Then they go backward, go back up to capture it. So the story here is that when the copepod try to, so first they detect these things because of the diatom chain is usually maintain a horizontal horizontal orientation, and this is probably difficult for the copper to actually capture it. So then they just think back. Because of the sinking flow will realign these things, make it vertically oriented. Then they go backward to capture it. So the thing is, I remember, the early work by Rudy Strickland and Gus Pfaffenhoff, they have observation on how a copper pod detect the capture a cilia diatom chain, but the copper pod actually are tethered. So when they are tethered, so they actually bring in this diatom chain repeatedly in order to feed on it. But here you can see that within 1 second, the copper pod can use their sinking motion to realign the diatom chain to make it in a good position to actually capture it. So you see that this kind of swimming behavior actually is a facilitator to actually capture a long diatom chain. And here is the flow. You see this is a micro PIV measured flow. You see that when they swim, they will sink back. This flow actually can rearrange, say reorient it, because of the flow direction, can reorient to make the chain become vertically oriented. So I see that here, this is also a behavior that are driving by the small scale fluid mechanics, which are actually for feeding purpose, not for the mechanical energy efficiency things. So by talking about so many different examples, I just want to show some discussion points. So I think my study here is guided by a book by the Tübingen's book, The Study of Animal Instinct. So what we see here, all this behavior, you can understand them. They are actually the innate behavior. So what the innate behavior means they just born with it, not actually learned. So here we see that all these innate behavior that observed for marine plankton are remarkably adaptive and significantly enhance their fitness. Also, those innate behavior, they actually undergo different types of convergent evolution in different species. And you see that in some closely related species, you see also there is a divergent evolution because of they have evolved into different paths. So those kind of things, you can see both those convergent evolution and also divergent evolution. Also, you see, we have to emphasize that because of although those marine plankton, they are morphologically, physiologically, and genetically very diverse, and they actually was governed by the same small scale fluid mechanics. So this obviously they will give some of the to drive some of the convergent evolution of their key behavior traits. So I just have to emphasize these fundamental rules played by small-scale fluid mechanics. That is, why do plankton they behave the way they do? You have to consider all these fluid mechanics effects in here in order to actually understand why do plankton behave the way they do. So the next one I want to say that this slides actually show my research theme. Basically is a combined, so we have a fluid mechanics sites at here, and it actually controlled by the low-Reynolds-number fluid mechanics or the small-scale diffusion, and also they actually determine this transport of mass and momentum to and from individual plankton, and the individual plankton by their side they have a different morphology and behavior, perception, response, and interaction. So these two interface each other will generate basically our fantastic marine organism, marine biology and evolution, this kind of stories here. So I just want to emphasize this is my research theme here. Yeah. So I have been working with many great scientists in this field, and I want to thank you for them, and specifically like Matt Johnson at WHOI, and also I want to thank Thomas Kilbo and Gus Pfaffenhoff, Rudy Strickland, and many other people that I work with during these past maybe 20 years. Yeah, thank you so much. So this is the papers that I have been talking within these things. Yeah, thank you.
Thank you very much, Houshuo. This is a fantastic talk and very fascinating questions and yeah, observations. So please, people in the audience, if you have questions, just go ahead. I will start maybe for a small question, quick one. Is there in this convergence evolution you are talking about that is governed by the fluid dynamics, can you put a size limits or size boundaries for these to happen, or for certain groups of organisms?
So according to my current study, so I'm actually talking about two processes. One is if the organisms are able to overcome the diffusion time scale. The other one can overcome the viscous time scale, right? These two parts. I can see that is say at the copper size is probably smaller than like 5 millimeter, those kind of range, right? And for the smaller range, that is probably like 20 micron. So it's a 20 micron to 2 millimeter, those kind of range. That was what I'm talking. Because I have no data at beyond those two ranges. I don't know. Because of especially there is an interesting part is these these cryptophytes, or these smaller cells of the algae cell. What they do, I haven't yet have enough data to show those kind of patterns. But why I can say that within the microplankton and also the mesoplankton, I think what I'm talking is actually a very powerful driving force that is determining their evolution of behavior, especially the behavior they do. Yeah, thank you. This is a great question. Thank you.
Thank you. Any other questions from the audience? I may ask another one. I have tons of questions about this,
okay,
because it intersects with some of my interests. So when we are talking about this convergent evolution or divergent evolution, even in the example, what was the name? The Mesodinium?
Yeah, Mesodinium, yeah.
Yeah, in Mesodinium. So you showed a number of different species that have different shape or jump limitation, which reminded me of a little bit of the Darwin finch, of the specialization for different seeds, etc. So this is, when you see this, do you think that this divergence is occurring on a short time scale, or it can take a long time scale in order to or they can switch even between like behaviors, like if let's say they are phototrophic and then some other heterotrophic?
I mean, I actually don't know the time scale of how those evolution happens at this level, but what I see that is because of they have a I mean, originally there's some ideas that they are actually probably the same thing in the very beginning. Then they have a diverge because of the shape changes, and they have different phototrophic out of I mean, different, say, for photosynthesis, some do the capturing, the heterotrophic stuff, right? So because of, if you have a photosynthetic, then you have to jump. So the jump, in order to make your fast jumping, in order to overcome a diffusion limitation, then your body will have one type of shape, and you probably by doing this, you sacrifice how you capture the prey, right? So but you still have to capture prey. But the capture process probably are relatively less efficient than those specialized in terms of capturing prey. So I see that because of the different partitioning in terms of their capability in the trophic modes, right? They diverge in different things. But we also see that even at one phototrophic, this photosynthetic dominant Mesothelium rubrum, you see that, those are all around the world. The species are distributed around the world, and there's actually a species complex. They actually still, you see that they converge. So there are some at a species level, they converge to something, but at the family level, they actually, because of different trophic modes, they diverge. So, but I don't know the time scale how those things happens. I still probably need to talk with somebody about these things. I think this is a great question. Yeah.
Thank you. Do you think is there a way to distinguish the selective pressures from the fluid dynamic itself versus this feeding behaviors that you observe, or the feeding choices? Because here there is the intersection between physical constraints, ecology and physiology, and I feel like it's a little bit hard to quantify in a meaningful way the contribution of each.
I totally agree. I actually don't have a clue how to actually disentangle it. So what I see is I measure their behavior. And I see the patterns. So the pattern is if you are phototrophic dominant, you jumping relatively fast at 6 body lengths. But if you are dominant as a heterotrophic, then you jump relatively slower and also jump irregular. But on the other hand, you can capture prey very efficient. So that's what I observe it. But I can only say that. I don't know how if people can design experiments to actually switch them between. I don't know. I mean, this is a very interesting question. I mean, if people can actually design experiments or do some kind of genetic things to modify or to identify what the genes actually control what things. I mean, this is very interesting. I see this Mesothelium complex family is a very interesting evolutionary process because of these are dominant one. These are very, very, you can get, say, you just go to the ocean, you can get them immediately like that. It's a dominant, it's a very widespread, and the shape are very, just like that, just converged to something like that. So I completely agree with, but my capability is to measure their behavior parameters. I see that's the difference. Yeah.
Thank you.
Thank you.
Any questions? Mimi, you are muted. Yes.
I have one. Great talk. Do you find these different feeding modes? Are the species that, for example, are cruise feeders versus filter feeders, do they tend to, I'm thinking of, are they better at capturing certain kinds of small prey? So, for example, are the filter feeders better at catching very small particles or bacteria, and are the ambush feeders better at catching modal prey? And have you looked at the patterns of where they occur and what prey are available?
I just started. I mean, this is a great question. I mean, the question is, I haven't yet started to look at these different prey type. What I see is that, like the two, say, for example, the tintinids and the strobilium, that's the cruise feeding versus the feeding current feeding. They actually capture the same inert particles. So like the cell, they don't move.
Yeah.
So those two things actually they capture same thing. But the other one, like ambush feeder, they need to capture those cryptophytes which are move.
Yeah.
They're swimming. So what I see here is that I can see it is all because of the, say, probably the tintinets. They actually, they just have a feeding current generated, so they probably live in a relatively food rich environment. So a lot of particles, they can just mooring around to capture it, right? To capture it. But the other one is probably if you're swimming very fast, you actually can encounter particles more. So probably they are in a relatively food dilute environment. And this is also for copepods is also true. Like a copepod, like a Classocananus, they is a truly cruise feeder. They're actually in the oligotrophic environments. So their food particles also very dilute. And also these two things, the cruise feeding, they're swimming very fast. They can encounter much more food. The encounter rate are high. So I just see these two feeding current and feeding current is more energetic efficient, but the encounter rate are relatively less.
And then I also wondered if any of the things that you've looked at, because we know that resources are very patchily distributed. So even though it's dilute, there are local regions where prey are more dense or less dense. And have you seen with any of your creatures that they change their behavior to being good swimmers if it's really dilute and they're trying to find a patch, and then change their behavior once in a patch?
I haven't yet done that. I'm actually trying to think about those kind of experiments, how to actually see them. These are what I can do is actually really a preliminary, results about their feeding. I just see there is this kind of 3 different patterns. I mean, there's a one step from forward from Fincher's work. I mean, Fincher, we haven't yet know exactly how they do it. So we just see how they do it. But how to play with different particles concentration, that will be a great next step. Yeah, thank you very much for this question. Yeah.
Yeah, I was just going to say we've been looking at choanoflagellates, and they can switch their morphology depending on whether there are predators around that detect the flow field they produce or not, and depending on whether the prey are too dilute or the prey are dense. So I was just curious if that great suite of other types of creatures you've been looking at might show similar things. So I'll bug you by email later.
Okay, thank you. Yeah, great. Thanks.
One more question. If not, we will stop here. Okay. Thank you so much, everyone. And thank you very much, Houshuo, for this fantastic talk. And we'll see you next month then. Thank you very much.
Thank you very much for attending. Thank you.
Thank you.